First, we need to know what is the meaning of the term translocation. So what is translocation? It is the process of movement of materials from the leaves to the non-green tissues. Okay, leaves are the green tissues which can prepare their own food. So movement of the food which is solute from the leaves to the non-green tissues. What does the non-green tissues include? They include a root, they include flowers, fruits, etc. And some developing leaves also. These developing leaves also require the prepared food. Okay. What else we need to know? How are they translocated from the leaves to non-green tissues? They are transported via vascular system. Okay. Vascular system have two parts, xylem and phloem. Now we know that xylem transport water and minerals and phloem transport food, hormones and other substances. We will see in this chapter what phloem transports. So here what we are talking about, translocation of solute. What does solute mean? Solute means food, means glucose. So solute are transported via phloem, which we will study. Okay. So again, the terms we need to know in this chapter are source and sink. So source are the regions which can where the food is produced. Okay. So source is the region of production of food and sink is the region of utilization of food. Clear? So food is moved from source to sink. Clear? This we need to know. This is the basic idea of this whole chapter. Clear? So translocation of solute. Look here. What is translocation? Is the movement of photoassimilates through the phloem is known as translocation. Now these photoassimilates translocate from the area of supply which is known as source to the areas of metabolism or storage known as sink. They move through phloem. Phloem has several types of tissues. Among them one is sieve element. Okay, they move through the sieve elements of phloem. Okay. Now sieve element includes highly differentiated sieve tube cells, sieve tube elements typical of angiosperm and unspecialized sieve cells of gymnosperm. Very important. So sieve element include what? They include sieve tube element and sieve cells. Sieve tube element which is typical of which plants? Angiosperms. Sieve cells is typical of which plant? Gymnosperms. And this sieve element which include both sieve tube and sieve cell, they transport what? They transport solute. Remember that. This is very important. Question comes based on this. Now, I told that phloem have several tissues. They include sieve tube, sieve cell, companion cell, parenchyma cells, phloem fibers. Okay. So, this phloem tissue contains companion cells also and parenchyma cells also. So, the role of companion cell is wide in translocation. We will study about that. But the role of parenchyma cells is they store and release food molecules. Okay. Some phloem tissues also include fiber and sclerids. The role of fiber and sclerids are for protection and strengthening of the tissues. Okay. Some phloem tissues also include latticifers, which are the latex containing cells. just for giving the general idea. Now let's solve one question. Which of the following option match the plant tissue with its correct function in vascular plant? So in the last slide only we studied that in gymnosperm it is sieve cell. Okay, so food conducting vascular system in gymnosperm is sieve cell. So 3 will be matched with D. So find out where is 3 is matched with D. This one. Okay, so in one go you have found out the correct answer. So let's check the another also. C4. Sieve tube element will be the typical of angiosperm. Just now we studied. Now tracheids is the water conducting element of angiosperm. So remember tracheids. Sorry, I said wrong. Tracheids is a water conducting vascular system of gymnosperm and vessel element is a water conducting tissue of angiosperm. Clear? Next we need to know in detail about the sieve elements. So sieve elements are what? They are elongated living cells. Okay, remember they are living cells. Sieve elements are elongated living cells. Okay, when these sieve elements mature, they lack several of the organelles which are mainly found in the living cells. Okay, they lack nuclei. When the sieve elements are mature, they lack nuclei, they lack tonoplast, microfilaments, microtubules, Golgi bodies, ribosomes. All of these organelles are absent in the mature sieve organelle. Okay, but these cells have a functional plasma membrane. The plasma membrane is functional, mitochondria is present, and plastids and smooth ER are also present. So please remember what are absent and what are present. Okay, plasma membrane, mitochondria, plastids and smooth ER are present. Okay, so the structure of this tracheiry element. So tracheiry element are what? They are the vascular system that conducts water. So these tracheiry elements and sieve elements have a difference. Okay, sieve elements conduct water, tracheiry elements sieve elements conduct food and tracheiry elements conduct water. But structurally, they have one main difference. That is, tracheiry elements are dead at maturity and lack plasma membrane and have lignified secondary cell walls. But sieve elements are living at maturity. Clear? So what we have to know from sieve elements, that these sieve elements are very elongated structure. They are connected end to end to form a tube-like structure. Okay, to form a tube-like structure. The sieve elements are able to live for a long time, as it is seen in palm trees that they are able to live for around 100 years. Even they have no nucleus, but still they are able to live for around 100 years. Okay. The cells closely associated with sieve elements. Okay. Another cells which are closely associated with sieve elements are companion cells. Okay, what we saw that the maximum of the organelles which were absent in sieve elements, it is present in companion cells. Okay, all the organelles are present in companion cells. So these companion cells provide the nutrients to the sieve elements and keeps the sieve elements alive for a longer period of time. Okay, the sieve element, and just giving you the brief idea so that You will have a conception to study the further topics. Sieve elements and companion cells are connected to each other via plasmodesmata. They are connected to each other by minute pores known as plasmodesmata. Okay, they are the common walls between the plant cells, which helps in the, they are similar to the gap junction in animals. Clear? Now, it is also found that the virus are able to enter the plants. They are able to move through these plasmodesmata. Okay, the virus can move through this plasmodesmata and they can enter into the sink tissues. Okay, so remember, viruses, they are able to move through plasmodesmata. Okay, where they will go? They will search for food. Where they will go? They will infect the sink tissues. What are sink tissues? Sink tissues are the non-green tissues where the carbohydrate is metabolized or stored. Clear? Okay, so if the virus enters the phloem, they will travel with the sap. Okay, they will travel with the sap, with the phloem sap, and they spread widely around the plant and they infect sink organs. But remember one thing, viruses are bigger and plasmodesmata is even smaller than the viruses. So to pass the plasmodesmata, they disassemble themselves. Okay, and once they cross the plasmodesmata, they reassemble. Okay, now we need to know about the girdling experiment. This experiment proves that phloem transports food. Okay, this is to prove that the phloem transports the food or the solute. So how to do that? What scientists have done, they demonstrated the transport of organic solute through the phloem. What they did, they removed the ring of a bark of a trunk of a tree. Now, removing the bark is known as girdling. Okay, so when a ring of the bark is removed, it was found that the sugar transport have been stopped from the leaves to the roots. Roots cannot prepare its own food, it requires the food from the leaves. So sugar transport from the leaves to the root have been stopped. But water transport was unaffected. So what does that prove? That organic solutes accumulate above the girdle, because if this is a leaf region, these are leaves and these are roots. Okay, and here the girdling occurred. So what happens? They cannot translocate downward. This here it's inhibited. Okay, they cannot transport. So in that case, there is an accumulation of solute in this region. Okay, solute is accumulated in this region and leads to the swelling in this region. Okay, so this experiment is to prove that the phloem is the one that will transport the solute. Now look here, this is a schematic diagram of mature sieve element. Okay, this is a single sieve element. Okay, this is the external view of the single sieve element, and this is the longitudinal section showing two sieve elements. Okay, this is one sieve element and this is second sieve element connected together by what? See, the pores in the sieve plates between the sieve tube elements are open. So these are what? See, I'll tell you. So these are what? See here. These are sieve plate. The pores are collectively forming the sieve plate. So look here. Sieve plate pore. So this is a sieve plate pore. This one. Okay. This together, this region is called as sieve plate. Okay. So there is a plasmodesmatal connections between the companion cell. This is the companion cell, and these are the sieve tube elements. So there is a plasmodesmatal connection between the companion cell and the sieve tube. There are several organelles present in the companion cell but absent in sieve tube. Okay, each sieve tube can be associated with one or more companion cells. So companion cell role is that they will perform the essential metabolic functions which the sieve tube element cannot do. Okay, so see, there are several organelles present here in the companion cell, but one specific kind of protein present in the sieve tube element is the P protein. We will discuss about that. Okay. Now what we need to know, the sieve tube elements and sieve cells characteristics. Okay, the sieve tube elements are found. Sieve tube elements are found in angiosperms and sieve cells are found in gymnosperms. How they look different? What are the characteristics that are different in sieve tube and sieve cells? So some sieve areas are differentiated into sieve plates. Individual sieve tube elements are joined together into a sieve tube. Okay, so what we found that here sieve areas are differentiated into sieve plates. So sieve areas are differentiated into sieve plate. There was the presence of sieve plate in angiosperm. Okay, and individual sieve tube elements are joined together. Okay, what we saw also that I'm drawing a rough picture that this is one sieve element and several of them are connected in this way to form a long tube. Okay, and these are what? These are sieve plate. So this is the characteristics of sieve tube and angiosperm. There are no sieve plates. All sieve areas are similar. Okay, there are no sieve plate. There is nothing, no sieve plate like this in angiosperm. Sorry, there is no sieve plate in gymnosperm. Okay, so all sieve areas look very similar. Now, sieve plate pores are open channels. Here, pores in sieve areas appear blocked with membranes. So, pores are not open. Here, pores are open channels which are conducted end to end to form a tube. Okay, so this is a sieve plate, the plate-like structure, and these are sieve pores, sieve plate pores. There are presence of P protein in angiosperm, dicots and monocots, but these P protein are absent in gymnosperm. Now, companion cells are sources of ATP and perhaps other compounds and are transferred cells or intermediary cells. There are different kinds of companion cells which we will study now, which are present in angiosperm, and there are albuminous cells sometimes function as companion cells in case of gymnosperm. So please remember this chart. This is very important. Now coming to P protein. So in the last slide we saw when we were studying about longitudinal section of the sieve tube element, we saw that in sieve tube element there is a protein named as P protein. What is P protein? The full form is phloem protein. What is the role of phloem protein? First of all, we need to know where are they found. They are found in angiosperm. They are found in many dicots, in all dicots and many monocots, and they are absent in gymnosperms. Very important too. They are not present in gymnosperms. They can be of several forms. They can be tubular, fibrillar, granular, crystalline, depending on which species they are present in and the maturity of the cell. Now, in immature cells, they are present as discrete bodies in the cytosol known as P protein bodies. Okay, in the immature cells also they are present in the P protein body forms, They generally disperse into tubular or fibrillar forms during cell maturation. Just a general idea. Okay, this is in general. What we need to know here that, wait, this is just a basic knowledge. What we need to know that P proteins are absent in gymnosperms. They are present in all dicots and many monocots. Now see, what is the role of this P protein? They helps in sealing of any damage. So what kind of damage will the phloem can face? The phloem are under high tension. Okay, they are under high internal turgor pressure. Okay, they are transporting the solute here and there. Okay, they are under high internal turgor pressure, due to which there can be damage to the sieve tube. Okay, due to which what happens, the solutes can be leaked here and there, and there is the extra loss of solute because of the damage or the injury in the sieve tube. Okay, so P protein, what they do, they seals off any damaged sieve elements by plugging the sieve plate pores. So see, if this is a sieve plate, okay, there are pores. So if there is any damage, what they do, see here, if there is a damage, they plug that damage, okay, they seals off that damage. When the sieve tube is cut or punctured, the released pressure, so when there is a cut or puncture, the pressure is released. Okay, high pressure is built up to transport long distance. Okay, when there is any cut or punctured, then what we will see, that the pressure is released, which cause the contents of the sieve elements to surge towards the cut end. Okay, towards the cut end, the solute will be leaked, which causes the plant to lose much of the sugar rich phloem sap, if there will be no sealing mechanism like that. So what do P protein do in that case? Whenever there is any kind of injury, this P protein and other cellular inclusions are trapped on the sieve plate pores. Okay, so if I change the color, so see, if there, here is the injury, if this part, red part, is the injury. Okay, in that case what will happen? The plug will be, it will form a seal. Okay, it will form a seal, and here the P protein will accumulate, which will form the seal. Including the P protein, there were other cellular inclusions which will be trapped in this region, so that the region is healed quickly. Okay, so that there is no further loss of the cell. Now the long term solution of this healing is of the sealing of the sieve plate pore is a deposition of callose. So what we studied that, if this is sieve plate, if here is a deposition of P protein, so how will the callose deposit? So the callose will deposit like, it will form the region hardened. Okay, the region will be hardened even further. Okay, so this is P protein, the callose will be deposited here around, and will even harden the surface, so that the sealing will be long term. Okay, thus the sealing will be long term. Now what this callose are made up of, they are made up of beta 1,3 glucan, and it is synthesized by the enzyme in the plasma membrane, and they are deposited between the plasma membrane and the cell wall. Okay, these callose are synthesized in the functioning sieve elements in response to several conditions, like in response to damage or other stress such as mechanical stimulation, high temperature, or the preparation for normal developmental events such as dormancy. Okay, dormancy, the seed coat needs to be hardened, so there can be the deposition of callose. Okay, So these are the role of callose and pea protein in the plant. Next, we will solve one question. Now, sieve elements of phloem conduct sugars and other organic materials throughout the plant. The following statements were made characteristics of sieve elements in seed plants. Angiosperm contains sieve plate pores. So just now we discussed, yes, they contain sieve plate pores. There are no sieve plates in gymnosperm. This is also correct because we studied it's not present. Sieve cells are present in gymnosperms. Okay. Pea protein is present in all eudicots and many monocots. Yes, it is absent in gymnosperm, but is present in all eudicots and many monocots of angiosperm. There is no pea protein in angiosperm. This is wrong. Pea protein is specific in angiosperm. Okay, so A, B, C are the right ones. This is the correct answer. So when the callose is deposited, it efficiently seals off the damaged sieve element from the surrounding intact tissue. So as the elements recover from the damage, the callose disappears from these pores. They are not permanent. It disappears from the pores when the sieve elements recover from the stress or the injury. Okay. Next we come to companion cells. We discussed that attached with the sieve element are one or more companion cells, and companion cells are connected with the sieve elements with plasmodesmatal connections. These plasmodesmata are often complex and branched on the companion cells. So see the picture of it when we will do the pathways of translocation. And these companion cells have dense cytoplasm, mitochondria, nucleus, Golgi, ER, and chloroplast. So all the organelles are present here in the companion cell which are absent in sieve elements. So there are numerous mitochondria present in the companion cell. So what is that role? That they constantly supply energy as ATP to the sieve element. Okay, they supply energy to the sieve element. They play an important role in the transport of photosynthetic products from the producing cells photosynthetic product from the producing cells which are the leaves, okay, to the sieve elements. Okay, they transport the photosynthate, that means glucose, from the producing cells which are the leaves to the sieve elements in the minor veins of the leaf. Clear? Now we also need to know that there are three different types of companion cells: ordinary, transfer and intermediary companion cells. All the three companion cells have dense cytoplasm and abundant mitochondria. Now look here how the three are different from each other. So ordinary companion cell have a peculiar characteristic that the thylakoid is well developed. Okay, please note down all this in your copy to have the idea that they have well developed thylakoid. Now we will do a comparative study. Okay, instead of going one by one, we will do a comparative study. Okay, ordinary companion cell have well developed thylakoid, but intermediary cells have poorly developed thylakoid. Okay, ordinary companion cell have smooth inner cell wall, but transfer cell have ingrowths, finger-like ingrowths in their cell wall, which increases the surface area of transfer. Okay, so the name is transfer cells, which have finger-like ingrowth in their cell wall, or the walls are folded, which increases the surface area of the plasma membrane, allowing greater transport. Okay, here intermediary cells also have smooth cell walls. Please remember what we discussed. The first point we discussed about the walls. Okay, we discussed about thylakoids comparison in ordinary and companion cell. Third, that there are relatively few plasmodesmata. Very important. Very few plasmodesmata are there in ordinary companion cells. Okay, They are connected only to their own sieve plate. They are not connected with the producer cells. Okay, these companion cells are not connected with the producer cells. They are connected with their own sieve element only, because plasmodesmata are very few. Now what about transfer cells? That these transfer cells peculiar characteristics is they have a finger like polyp. Okay, now one again important thing to remember, that these ordinary cells and transfer cells, they they take up the solute from the apoplast or the cell wall space. Okay, they take up the solute from the apoplast, but intermediary cells take the solute from the symplast. Okay, they transport the solute in the symplastic pathway. So where from they will transport? Every of the companion cell will transport the sugar from the source to the sieve elements. So what is the source? Source is mesophyll tissue of the leaf. Mesophyll tissue of the leaf. Okay, and it's transferred to the vascular bundle where sieve elements are present. Okay, from the sieve elements, this food will be translocated to the different. Okay, so in the plants where no apoplastic state appears, then there only intermediary cells work. Okay, because intermediary cells transfer the solute in the symplastic pathway. Symplastic means cytoplasmic pathway. Okay, so the patterns of translocation from source to sink. We know that the food is synthesized in the source and they are transported to the sink where they are utilized or stored. Okay, source include the exporting organs, typically mature leaves, which produce the photosynthesis in excess of their own needs. Sometimes the storage organ also serve as the, so they are either the mature leaves, and sometimes the source are also storage organs. Okay, so like wheat, carrot roots, cotyledons and endosperm of seeds, they also serve as a storage, they also serve as a source sometimes. Okay, so if I take one example of the storage root of wheat, scientific name is Beta maritima, what we see that in the first growing season, the storage root of wheat is sink. Okay, in the first growing season, they accumulate lot of food from the source leaves. So they are taking the food from the source leaf. They are acting as a sink. Okay, in the first growing season, but in the second growing season, the same root becomes the source. The same root will help the sugar present in it to remobilize, and all the sugar present in the sink in the second growing season, they will be used up to produce a new shoot. So in the second growing season, the beetroot's storage root will serve as a source where all the sugar present will be mobilized, and it will help in the emergence of the new shoot. Okay, so sometimes the source, sometimes the sink can also serve as the source. Okay, this is what we need to know. So what are sinks? Sinks are any non-photosynthetic tissues which do not produce any enough photosynthate for their own use or storage needs. They are dependent on the source for their food. They either cannot synthesize, or when either they cannot synthesize, or if they synthesize, they are they cannot meet their own demand. Okay, so sink can be roots, sink can be developing leaves. So developing leaves are green in color, but they cannot synthesize enough photosynthate. Okay, they require large amount of photosynthate, but they do not synthesize enough photosynthate, so they are dependent on the source. So since they are dependent on the source, they are also the sinks. Developing green leaves are also sinks. Okay, another sink include the roots, tuber, developing fruits, immature leaves, etc. Okay, so what we need to know that the source and sink can be reversible. Sometimes the sink also serve as the source. So according to the need of the plant, the source and sink can be reversed. Okay, according to the need or according to the season, they can be reversed. Like according to the season we studied in weed food. According to the plant need, say for example, if the plant is under any kind of stress, then what happens? That plant will mobilize or break its stored food to combat with the stress. Okay, so what we need to know that the movement in the phloem can be upward or downward. The movement in phloem is bidirectional. Okay, it is bidirectional. That is, the growing leaves are located towards the top and the roots are located towards the downward side. So the leaves also transport the sugar towards the upper side, which are the developing leaves, and also transport the food to the lower side, which are the roots. Okay, so they can transport the solute or the photosynthate in upward and downward direction. So they are bidirectional. Okay, or we can say they are multidirectional because they also transport the solute towards the lateral side also. Okay, so several factors we need to know which follow the source sink pathway. Okay, so proximity. So whichever leaves, so see here the larger leaves are there and here the developing leaves and the shoot tip is there. Okay, so whichever leaf is closer, okay, will provide the photosynthate to that sink. Whichever source is closer to the sink, that source will supply the photosynthate to that sink. So what I meant, for example, the upper mature leaf, like for this one. So look here, the upper mature leaf like this one, they will transport the photosynthate to the growing shoot tip because it is very nearer to the shoot tip. These two are very nearer to the shoot tip, so they will transport the photosynthesis to the shoot tip. Now the lower leaves will transport the photosynthesis to the root because they are nearer to the root. Okay. Now there will be some intermediate leaves also which export in both directions. They will transport upward also. They will transport lower side to the lower side also. Okay, intermediate leaf if there any, they'll transport both sides. Okay. Another see, according to the development, what we see that the shoot apex and root apex they serve as a major sink during vegetative growth because they require large amount of photosynthate during the vegetative growth. Shoot and root growth takes place during the vegetative growth. So the sink, like major sink, are the shoot apex and root apex during the vegetative growth. But during the reproductive growth, when the fruit starts developing, then fruit becomes the major sink. Okay, so what we need to know that in the vegetative stage, shoot apex and root apex are the major sinks, but in the reproductive stage, fruit is the major sink. Okay. Now, vascular connection, source leaf preferentially supplies sinks with which they have direct vascular connection. So, vascular connection. So, if this is a leaf, okay, this is again a developing leaf. This is again a leaf. Okay, so here the vascular connection is this way. Okay, so what we see that there is no direct vascular connection between the leaf 1 and 3. Okay, so if one is a developing leaf, then 2 is directly connected. 2 is if it's a matured leaf. So 2 is directly connected with 1. instead of three is not directly connected with one. So it's saying that the vascular connection, the leaves which have direct vascular connection will take the photosynthate from that. Okay, so preferentially zinc will take the photosynthase from those regions where they have direct vascular connections. So see, they modify their translocation pathways. So when there is any kind of wounding or pruning, what happens? That the direct connection between the vascular tissues is lost. Say example, if this is a plant and the leaves towards the left side fall due to any reason. Okay, see, or I can take one example that these lower leaves used to transport the solute to the root, but due to any reason the lower leaf fall off. If I think of a situation where the lower leaves fall off, so what will happen? Now the upper leaf will transport the solute. In that case, the upper leaf will transport the solute to the root. Okay, so this is known as anastomosis. Okay, so in absence of direct connection between the source and sink, their vascular interconnection anastomosis. That means they provide the alternative pathway of transport. So say for example in sugar beet, when the source leaves are removed from one side, the plant can bring about cross transfer of photosynthate. The photosynthate will be cross transport from another region. Okay. So if these leaves are removed, which used to transport the which used to translocate or which used to transport the solute to the upper side of this region only. So when these leaves are removed, so the leaves on the other side will translocate the solute to the upper side. Okay, so if any region like lower leaves are lower leaves, since it is closer to the root, they will transport the solute to the root. If lower leaves are removed, the leaf from the upper side will transport the solute to the root. Similarly, if upper leaves are removed, then lower leaves are forced to transport to the root also and to the growing tip also. So this is known as anastomosis, where alternative connections develop to transport the solute. Clear? Next, we need to know the materials translocated in the phloem. So sugar is the main material which is translocated in the phloem. The sugar is the sucrose, and this sucrose is a non-reducing sugar. This is less reactive, or this is unreactive. Okay. Another important compound which is translocated in the phloem is nitrogen, especially in their amino acid and amide form. In the amino acid form is glutamate and aspartate, and in the amide form is glutamine and asparagine. In this way, they are transported in the phloem tube. Plant hormones are also transported in the phloem tube. Nucleic acid and proteins, nucleic acid, nucleotide phosphate and proteins are also transported. Okay. Nucleotide phosphate is like NAD, NADPH. I said nucleic acid. It will be NAD, NADPH. Okay. P proteins are also translocated. Several proteins like chaperones, protease inhibitors, all these are also transported. Some inorganic ions are also translocated. But nitrate, calcium, sulfur, and iron, since they are immobile, they cannot be transported via fluid. Okay, remember the question asked from calcium is very common, that calcium is an immobile substance, immobile mineral. Okay, so they are not translocated by food. Now we know that sucrose is the most abundant kind of sugar translocated in the phloem. Now some plants transport the sucrose in the polysaccharide form. Okay, oligosaccharide of raffinose series. Okay, some plants transport oligosaccharides also. These oligosaccharides belong to the raffinose series. So what does raffinose series include? Number one, they include carbohydrate which contains sugar, plus varying number of galactose molecules. So if I take under raffinose series, number one, raffinose. So these raffinose, what they contain? They contain one sucrose and one galactose. So Phloem also transport oligosaccharide of raffinose series, which under raffinose series, raffinose is one which contains one sucrose, one galactose. Next is tachyose, which contains one sucrose and two galactose. And the third is verbacose, which contains one sucrose and three galactose. So these are more bigger than the sucrose. Okay, there are some alcohols which are also translocated, which include mannitol and sorbitol. Now the sucrose which is transported, it is non-reducing sugar. Okay, the glucose and fructose are the reducing sugars. These reducing sugars have an exposed aldehyde or ketone group. So when an aldehyde or ketone group is exposed, they become very reactive. So if they become reactive in the translocation pathway, in the translocation tube, then what we see that it can react with several other proteins and any metabolic substances present in the tube, which is again very harmful for the plant. So we need some, so the plants need some non-reducing sugar to be transported. Okay, so they transport sucrose. In sucrose, the ketone and aldehyde group is reduced to an alcohol. Either they are reduced to alcohol or combined with similar other group. Okay. Now the non-reducing sugars, since they are less reactive, they do not show any hindrance to the flow of the sucrose. Okay, they do not show any synthesis of any toxic compounds or any unwanted materials in the phloem tube. So this is the most commonly translocated sugar. So which of the following compounds is generally translocated in the phloem? We already discussed, it is sucrose. Okay. The most important topic of this chapter is the pressure flow hypothesis. This was proposed by E. Munch in 1926 and it is the most accepted mechanism of phloem translocation. This is the most accepted mechanism for phloem translocation. Now, this hypothesis explains that it is because of the pressure which is generated in sieve elements and companion cells which drives the transport of solute from the source to the sink. Okay, so the movement or the transport of the food from the source to sink is a long distance transport. Okay, and this transport is due to the pressure generated between the sieve element and companion cells, which is the source weight. So it is the pressure gradient between the source and sink drives the movement of the food from the source and sink. Okay, so in leaves, sugar is synthesized in the mesophyll cells, and from the mesophyll cells, they are pumped into the phloem using the metabolic energy. Okay, the pumping of sieve, the pumping of sugar into the phloem is driven by metabolic energy. So it is said that by using the energy, the sugar is not only transported to the phloem. Wait. The sugar is not only transferred to the phloem but it is also concentrated. Okay, it is said to be as the sugar is also concentrated when it is transferred to the phloem. So see, when a solute such as sugar is concentrated inside the cells, water enters the cell. So see, this is a source which are the leaves when the when the solute, which is the photosynthate, or the when the sugar is accumulated in the sieve tube element. This is a phloem, sieve tube element of phloem. So when the sugar is accumulated from the source, intermediate is a companion cell. When they are accumulated in the sieve tube, then what we see, the water enters in. Okay, the sugar has entered into the sieve tube as well as the water has entered the sieve tube. Here, now, since the cell wall of the plant are very rigid, okay, so since the water has also entered and the solute has also entered, there is high internal pressure created in the sieve tube. Okay, this pressure causes the sap to move through the pores down the tube. So these are the pores. These are the pores. So this sap, due to high internal pressure, the solute is moved to the downward position. Okay, the sap is moved through the pores to the downward position, and the downward is located in the sink tissue. So see, at the other end of the transport stream, where the sinks are located, sugar constantly leaves the phloem, and when they are leaving the phloem, they are either used by the surrounding cells. Okay, some of them are consumed as an energy source while they are moving from the while they are moving into the tube. Some are used as an energy source. Some are stored as sugar in the sink. Okay, and some are used to make new cells if the sink tissue is growing. So this is a fate of glucose, or the sugar, not the glucose, is a fate of sucrose in the sink tissue. Okay, since sugar leaves the phloem in the sink, now one more thing, see, now here what we see, that sugar is leaving the phloem tissue and it is entering the sink cell. So when they are entering the sink cell, see, the water here is leaving. When the solute, or when the sucrose is entering the sieve tube, so when I say sugar, when I say solute, when I say sucrose, they all mean the same thing. Okay. So see, the solute, when enters the sieve tube, then water also enters. When solute leaves the sieve tube, then the water is also released back. This is the xylem from where the water comes and goes away. So when the water enters the when the sugar enters the sink, the water exits too. Okay, when the water is released, the water goes back to xylem. In that case, pressure goes down. Okay, so due to this pressure gradient, it helps in the transport of sugar from the source to the sink. This process is known as pressure flow mechanism. Here, so again I summarize with a different picture. So see here, in the first case, what happens? This is xylem, this is phloem, this is companion cell and source cell, this is water and sink cell. This is again companion cell, and this is sink cell, which can be root, developing leaves. Okay, so look here. So in the first case, what happens, that active phloem loading into the sieve elements. This is a phloem. Phloem is loaded. Okay, two terms we need to remember: phloem loading, which I'll explain in detail in the next few slides. Phloem loading and phloem unloading. So in case of phloem loading, what happens is the phloem is loaded with sugar. So where is the sugar coming from? Sugar is coming from source. So phloem is loaded with sugar. So when the phloem is loaded with sugar, water also comes in during that case. So which builds up the turgor pressure over here. Due to high pressure, this helps the movement of solute downwards. Okay. So see, the pressure driven bulk flow of water and solute from the source to the sink. Now once they are near the sink area, once they are about to enter into the sink, the water will be the water will be lost. The water will move back to the xylem. Okay, so this, the water is leaving the phloem. So this is phloem unloading. Wait, the water is leaving the phloem, and the water and the solute is entering the sink cell. Okay, since the solute is entering the sink cell, we can say that the phloem is now unloaded. When the solute is coming to the sieve tube, we say phloem is loaded. When the solute is going away from the phloem, we say that phloem is unloaded. Okay, so active phloem unloading, in that case, when the solute is moving towards the sink cell, then water goes out. Okay, so turgor pressure in that case is decreased. So again I have explained that sugar is actively loaded into the sieve element. When the sugar is loaded into the sieve element, water enters the phloem cells, which builds up the high turgor pressure. And at the sink, the sugar is unloaded. Okay, so towards the source region, the sugar is actively loaded. Towards the sink region, the sugar is unloaded. In the loading condition, what we see that sugar enters the phloem cells. When the sugar enters the phloem cells, water also enters. When water also enters, this builds up the turgor pressure. In the sink, near the sink, what we see that the phloem is unloaded. That means the solute enters the sink. When solute enters the sink, the water leaves the phloem. So the turgor pressure is lowered down. Okay, so water together with dissolved solutes, they move by bulk flow from the area of high pressure, which is near the source, because water and solute are together present in that area, creating large tension, creating huge turgor pressure. So water and solute will move from the region of high turgor pressure to the region of low pressure. Clear? So whenever there is the accumulation of sugar in the sieve elements, that is phloem loading, since the sugar concentration is very high, water potential will be dropped. We all know when the concentration of sugar is very high, water potential will drop. In that case, water will enter the sieve element and causes the turgor pressure to increase. We are just, I'm explaining it in different words. Okay, so that the concept will be crystal clear to all of you. Now in phloem unloading, what we see that the solute enters the sink and it leaves the phloem, which is phloem unloading. So since the solute is entering the sink cells, water will leave the sink tissue. Sorry, water will leave the phloem tissue, which will lower the osmotic pressure within the sieve element. So thereby what we can see therefore that pressure gradient is built up from the source to the sink, which helps in the transport of sugar from the source to the sink. Okay, this osmotically generated pressure gradient leads to the bulk or the mass flow of materials between the source and the sink. Clear? Next we need to know in detail about what is phloem loading and unloading. So remember one thing, when the solute is transported from the source to the sieve elements, okay, I'll show you the picture in the next slide. Remember one concept that when the solute is transported from the source where it is produced to the sieve elements, it is known as phloem loading, means phloem is loaded with the sugar now. When from the sieve elements they are emptying into the target cells. So the movement from the sieve elements to the target cells is phloem unloading. That means that now the phloem becomes empty because it is transporting the solute to the target cells. So it's phloem unloading. Now phloem loading and unloading both shows apoplastic and symplastic method of transport. Okay, both loading and unloading occurs through apoplast and symplastic pathway. Now look at this picture. Wait. In this picture, see, these are mesophyll cells. The first few cells are mesophyll cells. The carbon dioxide is fixed and the sugar is produced, which through plasmodesmata, it will enter into the bundle sheath cells. So what is the pathway we will write? From mesophyll cell to bundle sheath cell. After bundle sheath cell, they are entering into phloem parenchyma. So first we are looking this part. First we are looking this part. Okay. So see, sugar is moving from a mesophyll cell to bundle sheath cell to parenchyma and then to the companion cell. This is a companion cell. Then to companion cell and then into the sieve tube element. Okay. So see here, the cytoplasm is involved in the transport and the transport is through plasmodesmata. This pathway is known as symplastic pathway of phloem loading. Clear? The symplastic pathway of phloem loading is energy independent. Clear? Now look here towards the left side. Here what we see that from phloem parenchyma, see there are no plasmodesmata over here. So from phloem parenchyma, the sugar will come to the apoplastic region. From the apoplastic region, they will be pumped inside the companion cell. Okay, to pump the sucrose inside the companion cells, protons are required because they will be pumped by the help of a transporter, sucrose H plus transporter. Okay, this will help in the pumping of the sucrose to the companion cell, and this companion cell will transport the solute to the sieve tube element through the plasmodesmata. Okay, so here the plasmodesmata is here the companion cell is the type of companion cell here is the transfer cell. Here the type of companion cell is transfer cell. So I discussed that intermediary companion cells have large number of plasmodesmata and transfer cells have fewer number of plasmodesmata. They have the plasmodesmata mainly connected with their own sieve element rather than the other cells. So they are not connected with the source cell. We discussed in the last few slides that they are not connected with the source cells. They are connected with their own sieve tube element. Clear? So sugar is pumped into the companion cell, then through the plasmodesmata. Then from the companion cells through the plasmodesmata, they enter the sieve tube element. Okay, this pathway is known as apoplastic pathway. Clear? And remember one thing very important, that when from the apoplastic region sugar is pumped, from the apoplastic pathway, when sugar enters the companion cell, they require energy. Okay. So in one step of the apoplastic pathway, energy is required. So what is the pathway? The pathway is from mesophyll cells to bundle sheath cells, from bundle sheath cells to phloem parenchyma, from parenchyma to companion cells, from companion cells to sieve tube element. Sieve element, okay? Now look at this picture. The first pathway we discussed was totally symplastic pathway. Okay, in symplastic pathway what we saw that the sugar is translocated through specialized companion cells known as intermediary cells which has large number of plasmodesmata. They enter all the way from mesophyll to the sieve element. Okay, they do not enter any apoplastic pathway. They enter all the way from mesophyll to sieve element. In the second pathway, which is partly apoplastic. Why partly apoplastic? Because after some time, they again enter into the cytoplasm, which is there that they again follow the symplastic pathway. So at one step only they follow the apoplastic pathway. At one step only they are entering into the cell wall. Okay, then they are following the symplastic pathway. Clear? There can be the chance that they move through the apoplastic pathway to the sieve element also. Okay, so here what we see, in the initial few steps they are following symplastic, then in one step they are following apoplastic pathway. So we cannot say that they are completely apoplastic. They are partly apoplastic in this region. Okay, this apoplastic pathway, they will move, or they will look here. So in the apoplastic pathway, from the mesophyll cells via the bundle sheath cells, they enter the extracellular apoplastic region. In the apoplastic region, by the help of active transport, they enter into the companion cells. Now here the companion cells involved are transfer cells. Okay, these companion cells do not have the connection with the source. These transfer cells have no connection with or no plasmodesmata with source, but have with but have plasmodesmata with sieve element. But they have plasmodesmata with sieve element. Okay, so ultimately via the plasmodesmata they enter into the sieve tube element. Clear? Now the loading of sucrose and amino acid from the apoplast. So I told again, from the apoplast to the sieve element. Wait. So from the apoplast to the sieve element companion cell complex, they require what? They require H plus ATPase. Okay. So I can present in that way also, that if this is if this is the apoplastic region, so if we consider that this is the region, this is the apoplastic region, and the sucrose needs to be transported inside the companion cells. Wait. So sucrose needs to be transported inside the companion cell. How they will enter? Because see here, there are no plasmodesmatal connection. Okay, this companion cell is a kind of transfer cell where they have no plasmodesmatal connection with the source cell. So how they will enter, how the sucrose will enter the companion cell? They will enter by a pump present in this region. So see, in this region, I have elaborately drawn here that there is a pump present in this region, that the protons will come in the apoplastic region. Wait. Okay, so see, proton will come in the apoplastic region and via the sucrose H plus symporter. This is sucrose H plus symporter. Via the sucrose H plus symporter, the sucrose will be transported inside the companion cell with the help of proton. Okay. So see here, this is sieve tube, sieve element companion cell complex. This is the apoplastic region. Okay, this is an apoplastic region. So here the pump is present which helps the transport of sucrose inside the companion cell. This is a companion cell region. Okay, so at first the proton enters inside the apoplast. Okay, this is apoplastic region. And where the sucrose is high in the apoplast because they can enter the apoplast. So once sucrose is high in the apoplast, the protons come in and the protons are then co-transported with the sucrose inside the companion cell, where from the companion cell they will be transported via the plasmodesmata to the sieve tube element. Okay, via plasmodesmata. Now let's solve one question. Carbohydrates synthesized by photosynthesis are converted into sucrose and transported via phloem to other parts of the plant. The following aspects are associated with sucrose uploading in phloem and its transport. Both reducing and non-reducing sugar are transported. So we saw that only non-reducing sugar are transported. Reducing sugar are not transported. Reducing sugar example includes what? Glucose, fructose. They are very reactive. Okay, so they are not transported. But non-reducing sugar, which is most abundant in the phloem tissue, that is sucrose, they are transported. Clear? So first one is wrong. So when first one A is wrong, so this one is wrong, this one is wrong, this one is wrong. So which is remaining? BCD is correct. So see what is BCD? Sucrose uploading can be both symplastic and apoplastic. We read that uploading or loading, uploading means loading. Okay, so loading and unloading both shows apoplastic and symplastic pathway. The route of phloem uploading is mesophil. It can be bundle shell in between, okay, but the sequence is still correct. Phloem parenchyma, companion and sieve tube, correct. Transport in sieve tube is as per pressure flow model. Yes, this is also correct. So BCD is the correct option. Next, let's see. Given below are statements describing various features of solute transport and photoassimilate translocation in plants. Apoplastic phloem loading of sucrose happens between cells with no plasmodesmatal connection. So correct, apoplastic loading is where plasmodesmatal connections are not there. That's why apoplastic loading. This is correct. Growing vegetative sinks usually undergo symplastic phloem unloading. So vegetative sinks usually undergo symplastic phloem unloading. This is also correct. Movement of water between the phloem and xylem occurs only at source and sink regions. It is not necessary that movement of water between the phloem and xylem is only at the source and sink regions. It can be at anywhere. It can be in any region of the plant. So this is wrong. Symplastic loading of sugars into phloem occurs in the absence of plasmodesmata. No. Symplastic is in presence of plasmodesmata. Okay, so C, D are the incorrect options. So this is incorrect, this is incorrect, this is incorrect. So A, B is the correct option. Next, see. In this question is saying that which one of the following statement describes the process of phloem loading. So triose phosphate is transported in from the chloroplast to cytosol. No. Triose phosphate do not play into role into phloem loading. Sugars are transported. Sucrose plays the role. Sugars are transported into sieve element and companion cell, correct? Sugars are transported from producing cell in the mesophil to cells in the vicinity of the sieve tube elements. So it is saying that is it transporting from the mesophil to the cells in vicinity? Phloem loading means what is transported to the cells in the vicinity of sieve elements or in the sieve elements. Understand the question properly. The question is saying, are they transported to the cells in the vicinity of sieve element, or they are transported to the sieve element only? What is phloem loading? That sugars are transported from the source to the sieve element. Here it's saying it is transported to the cells in the vicinity of sieve element. In the vicinity of the sieve element are what? Bundle sheath cells, parenchyma cells. So transporting to bundle sheath cell and parenchyma cells is not the phloem loading. The transporting of sugar from mesophil to direct sieve element is phloem loading, not to the cells in the vicinity. Okay, that is just a transport pathway, but loading is when the sieve element is loaded. This is incorrect. Solutes are transported from roots to shoots. No, they are transported from source to sink. And root is not the source. Okay, so B is the correct answer. Another important concept we need to know, that see here the sucrose came from mesophyll cell to the bundle sheath cell. Okay. Now in the bundle sheath cell, from the bundle sheath cell they will be transported to the intermediary cell. Okay. So glucose are formed, like carbon is fixed and the glucose is formed. The glucose and fructose will combine to form sucrose. The sucrose will move to the intermediary cell. In the intermediary cell, what we will see, that this sucrose combines with variable number of galactose molecule and form raffinose. We studied raffinose series, raffinose, stachyose and verbacose. So depending on the number of galactose molecules they bind to, we can say that they are able to form different kinds of raffinose series of sugars. Okay, so they form raffinose. These raffinose, stachyose and verbacose, they are very large molecules. Okay, they cannot cross the they cannot enter the bundle sheath back. Okay, because they are very large molecules, they are not able to enter back to the bundle sheath cell, so they are concentrated in the intermediary cell. They remain concentrated to the intermediary cell. From the intermediary cell, these raffinose and stachyose are able to diffuse into the sieve element. Okay, they are able to diffuse forward, but they are not able to diffuse backward due to their larger size. So as a result, the concentration of the sugar rises in the intermediary cell and the sieve element. Okay, this is known as a polymer trapping model. Next, we need to know about the difference, just giving a brush up. We already know about the difference in the apoplastic and symplastic loading. So, apoplastic loading transport only sugar, but symplastic loading, since the cytoplasm is involved, other oligosaccharide in addition to sucrose are transported. So, for apoplastic loading, we know that companion cells are of ordinary and of transfer cell types. For symplastic, they are of intermediary cells which have large number of plasmodesmata. So the plasmodesmata in ordinary and transfer cells are few, and for intermediary cells the plasmodesmata are abundant. Okay. Now coming to the last part, phloem unloading. So phloem unloading also happens by symplastic and apoplastic pathway. So phloem unloading is when the movement of the solute is from sieve element to the sink tissues. Okay, in the symplastic pathway, what we see, that the sap reach the cells of the sink via the plasmodesmata, and in the second case it reaches the sink via the apoplastic pathway. Okay, the movement through the apoplastic pathway is an active process. We already discussed this. Now look here, pathways of phloem unloading. It can be symplastic and apoplastic. Now apoplastic pathway are of three types. The apoplastic unloading pathway are of three types. So symplastic is very simple, that they will enter into the sink tissue via the plasmodesmatal connections. Clear? Now in the apoplastic phloem loading, see there are three types: type 1, type 2A, type 2B. In type 1A shows apoplastic unloading. So see, in type 1A what we see, that in one step, in the initial one step, what we see, that transport from the sieve element companion cell complex to the successive sink. There is one step here which requires apoplastic pathway. Okay, so there is one step over here which requires apoplastic pathway. So from here it will move to the apoplast, and from the apoplast it will move to the symplast. So from the apoplast, when it enters the symplast, they require energy. Okay, so this is a very uncommon type of one in the phloem unloading. Now second, see, in type 2A what we see, that the apoplastic step occurs later in the pathway. See, apoplastic step occurs later in the pathway. Okay, and in type 2B what we see, that apoplastic step is further removed. It is symplastic sieve element unloading. Okay, next. Now the presence of plasmodesmatal connection ensures that the movement will be through symplastic pathway. Absence of plasmodesmata will show that The movement will be through apoplastic pathway. Now allocation and partitioning, another important term of this chapter, that the fate of the carbon fixed we need to know. What is the fate of the carbon fixed in the plant? So either they will be stored or utilized or exported. In the sinks, when there are the sugars present, they are used for growth processes or storage. Now another important term, allocation, is the fate of the carbon fixed. When they are allocated, how they are used in the plant. Now what is partitioning? Partitioning is the differential distribution of photoassimilates. How much of the photoassimilate will enter into the sink tissue? How much photoassimilate will each sink should receive? So that there will be the proper or equal size of the sinks. Okay, it's not that one sink is receiving more photoassimilate and another is receiving very less. There has to be equal partitioning. Okay, there has to be the partitioning. Now partitioning depends on several factors. Okay, so all sinks are competitive. So photoassimilate should be partitioned to all active sinks. Okay, so they depend on the three factors: the nature of vascular connection, how closer is the vascular connection of the sink to the source. Also they depend on the proximity of the sink, means how closely the sink are located to the source, how direct is the vascular connection, how closely are they located, what is the sink strength. Okay, if sink strength is high, then more amount of photoassimilate will be transported. So sink strength is a measure of the capacity of a sink to accumulate metabolites. Okay. Based on this, we will solve one question. The following are some statements about long distance translocation of photoassimilates in higher plants. So long distance transport is by pressure flow hypothesis. Okay, it is by pressure flow hypothesis, which involves phloem loading and unloading. Okay, which involves phloem loading and unloading. They are the long distance transport. One minute. So see, sugars are translocated in the phloem by mass transfer along hydrostatic pressure. So sugars are translocated in the phloem by mass transfer along hydrostatic pressure. Yes, the pressure is forming the gradient for the transport of sucrose. Gibberellic acid stimulates the unloading of sugar and phloem tissues and apoplast. We did not find any hormone playing a role in translocation. Munch pressure flow hypothesis is crucial to drive translocation and phloem. Yes, we studied that. Allocation and partition of carbon within the source leaf determine the phloem loading phenomena. Yes, correct. So B option is incorrect. So this are incorrect.